Now coming to the most important model, which always come for exam, okay? The nucleosome solenoid model proposed by Rombert and Thomas. Okay, it's the most widely accepted. So that's why they keep asking during exam, okay? Because this is the one the whole world accepts. So chromatin is composed of repeating units called the nucleosome, okay? Nucleosomes are the fundamental packing unit particles of the chromatin and give chromatin a beads on a string appearance in the electron micrographs. Okay. So here the basic unit of this model is your nucleosome. Okay. And these are the packaging units which give the appearance of the beads on a string. Okay. Like these, these beads, and then suppose this is a string. Okay, and you are wearing it around your neck. Okay, so let's move on. Okay, one complete nucleosome consists of a nucleosome core, linker DNA, and an average molecule of one H1 histone and other associated chromosomal proteins. So you have the core, the linker DNA, and the H1 histone. Okay. So remember I gave you to draw that because it always comes for exam. Usually you have to draw this table only. Okay. So the first level of packaging. Should I zoom in or is it enough? Not possible more than this, I think. Okay, let's just try and it's not going anymore zooming, so I'll just read. Okay, just listen. So there are many levels of packaging. 1, 2, and the final level. Okay, so there are 3 levels of packaging. So in the first level, the DNA is wound around a protein core to produce a bead-like structure called a nucleosome. Okay, this gives the packaging ratio of about 6. The structure is invariant in both the euchromatin and the heterochromatin of all chromosomes. The protein core is composed of 8 histone proteins, 2 of H2A, H2B, H3, H4. Histone H1 forms the linker between the 2 nucleosomes. Okay. 146 base pair of DNA is wrapped around each nucleosome. Okay. So the one that I gave you to practice, that is the easier one. Okay. But if you get time during exam, suppose it's open book exam, then you write this one from the note. Okay, write as fast as you can, squeeze as much as you can, because the more you write, the more we can give you marks. Not me, I mean whoever corrects your answer script, because looking at this condition, I think again it will be open book. Matlab, not open book but online exam, so you write this, or if you lack time, the one I have sent you for practicing also, that shorter one also will do. So here we are having a bead-like structure called the nucleosome, and the ratio is about 6. Okay, so so what it means here is that so this line packaging ratio of about 6, what it means is that let's say for example a naked DNA, okay, this is the normal naked DNA without any coiling and all. Suppose this one is 6 meters, okay, obviously it cannot be 6 meters, it will be 6 angstrom or whatever, but I'm giving you in the metric system so that you can understand. Okay, let's say it's 6 meters. Then after it coils, in that nucleosome coiling, right, in this model coiling, it became 1 meter. Okay, so the ratio means is 1 to 6. Okay, so let's say you are 6 feet tall, okay, you are 6 feet tall. Okay, you're 6 foot. Then I told you, Bandriti, coil yourself, make yourself small. Then you coil yourself. This is your legs, this is your arm, and you became only 1 foot tall. Okay, so that it means, that is what it means by packaging ratio. So I think it's clear. I mean, I gave so many easy examples. Okay, let's continue. So the protein core, the histone proteins, you already know, no? We already studied in the upper part. So this is just, they are telling you what it's composed of. Then here, these are the diagrams you have already drawn, no? These are your double helix, and then when they pack, suppose this one is 6 angstrom, this will be 1 angstrom, okay? It's packaging ratio of 6. Okay, then let's look at the second level of packaging. Then what happens is those coiling of beads in a helical manner, a helical structure, let me underline. Coiling of beads in a helical structure called the 30 nanometer fiber is found in both interphase chromatin and mitotic chromosome. This structure increases the packaging ratio to about 40. Okay, it means suppose I have what to say, a bike. Okay, let's say a scooty. I have a scooter and only I can fit. Okay, then I told girls, let's all fold ourselves. Okay, so Banriti folded, then Chaisa folded, then Riya, she folded, no, then Juhita also folded. Okay, then Mibisha also folded, no, all of you folded, no, and somehow in that one scooty 40 of us we can fit. You get it? So means in one, let's say in one cell, if this DNA is like 40 meters long, no, okay, in one cell 40 meters long, on folding it can no. See, in one cell means instead of if the DNA was just open, no, and just hang around like this, only one DNA will fit. But if you superfold it, you can fit do 40 units of DNA. Okay, so means obviously our DNA, it has so many genes, no, 10,000 genes I think. So you can imagine in one cell we can fit so many because of the superfolding. Okay, so continuing, this appears to be a solenoid structure with about 6 nucleosomes per turn. So thus gives a packaging ratio of 40, which means that every 1 micrometer along the axis contains 40 micrometer of DNA. So there it is explaining it very well. The stability of this structure requires the presence of the last member of the histone gene family, histone 1, because experiment that strip H1, histone 1 from chromatin, maintains the nucleosome but not the 30 nanometer structure. So H1 is important for the stabilization of the 30 nanometer structure. Okay, so here it's talking about this one. So on coiling, no, so we get this solenoid appearance. Okay, this solenoid appearance on coiling, and the length is about 30 nanometer. Okay, and to make this coiling of 30 nanometer diameter stable, we need that H1 protein. Okay, so let's look at the last one. So the final level, the fiber is organized in loops, scaffolds and domains that give a final packaging ratio of about 1000 in interphase chromosome and about 10,000 in mitotic chromosomes. So this fiber, it will arrange itself in loops, okay, in loops and scaffolds, okay, other sort of folding, so that we can package of about 1000 in interphase chromosome. Okay, so we can package 1000 times the size of that cell, okay, and 10,000 times the size of mitotic chromosome. Why? Because simply mitosis, the chromosome, they double, no, remember replication happened, so 10,000 like 2 times of the interphase chromosome, no, in one cell. Okay, so just imagine, okay, in a normal cell already this much, okay, 1000 times we are fitting. Then in the mitosis, not just 1000 times, double of this, okay, everything has doubled. Okay, so they have squeezed and squeezed and squeezed themselves so that they can fit in the mitotic chromosome. Then the final level of packaging is characterized by 700 nanometer structure. So this one, you can, if you zoom in, you can see the supercoiling. But you've already drawn, no? So you already saw it, okay. The condensed piece of chromatin has a characteristic scaffolding structure that can be detected in metaphase chromosomes, okay. So if you look intensely, if you really have a good a microscope with lots of magnification, even the one in our department, if you really look inside, you will see some sort of folding. Okay, so here this important part is done. I'll upload it separately. Okay, I'll upload it now.